One more. Okay, so just to check, can you hear me?
Yes, Doc.
Yes, Doc.
Yes,
Yes, Doc.
Thank you. So good morning, everyone. So for today, I'm going to present to you the microscopic or the histologic structures of the integumentary system. Okay, so let me share my screen. Okay, so for the integumentary system, this is composed of the skin and its appendages. Okay, so this is the largest, the skin is the largest organ of the body that covers the external surface of the body. So, okay, so the integumentary system, which is composed of, or the skin rather, which is composed of two main layers. Okay, the superficial, the outer layer, which is the epidermis. Okay, and then which is derived from the ectoderm. Then the deeper second layer, which is known as the dermis. Okay, this arises from the mesoderm. Beneath the dermis is this layer of which contains large amount of adipose tissue or the panniculus adiposus, and you call this layer as Ron.
The hypodermis, Doc.
Okay, so this is the hypodermis, otherwise known as the subcutaneous layer or the subcutaneous fascia. Although this is not part of the skin, but it blends with a deeper portion of the dermis, and it gives looseness and mobility to the skin. Okay, now histologically, we classify skin as to thick or thin, depending on the thickness of the epidermis. Okay, and usually thicker epidermis are those found in the palms of the soles and the palms of the hands rather, and soles of the feet. So these are the areas that are frequently subjected to friction, pressure, or abrasions. But anatomically, the thickest skin is found where? Where is the thickest part of the skin anatomically?
On the back, Doc.
In the upper portion of the back, Doc.
In the upper portion of the back. And which layer is thicker here? The dermis or the epidermis?
The dermis,
The
Doc.
dermis, Doc.
The dermis, Doc.
The dermis. Whereas the thickness of the epidermis in the upper portion of the back is similar to the thickness of thin skin found in other parts of the body. Okay, so we now go to the layers, the different layers of the epidermis. Okay, so what type of epithelium that lines the epidermis? What is this epithelium?
Squamous epithelium, Doc.
Come again?
Stratified
Stratified
squamous.
squamous epithelium, Doc.
Oh, why squamous? Not cuboidal.
Because the superficial surface stuff is a squamous.
Yes, so the superficial layers here are composed of flattened cells. Okay, so it is composed, this is one of the common example of stratified squamous epithelium, but here it is keratinized. Okay, the epidermis is avascular, so the nutrition here would be diffusion, by diffusion of substances coming from the underlying capillary network located in the dermis. So from the deeper innermost layer of the dermis, the epidermis rather, we have the stratum basale. So how do you describe this layer? Nika Pagador.
Cuboidal, Doc.
So it's composed of how many layers of cells?
Usually one, Doc.
Okay, so this is single layer of basophilic small cuboidal or low columnar cells which rest on the basement membrane. Okay, and what is the distinguishing feature of your stratum basale?
Germination, Doc.
Come again?
Mitotic division.
Okay, so there is intense mitotic activity present in this layer, and together with the initial portion of your stratum spinosum, the adjacent stratum spinosum, this layer are responsible for the constant reduction of epidermal cells or keratinocytes in order to replace dead cells that are lost on the surface of the skin. Okay, so this contains the stem cell of the epidermis. So that is why your stratum basale is otherwise known as stratum germinativum. Okay, so this is for the epidermal cell renewal. So the predominant cells present here or present in the epidermis are your keratinocytes. Okay, within the cytoplasm, it may contain melanin granules. Okay, so this melanin granules comes from your melanocytes, the pigment-producing cells of the skin, okay, which are also located, which are found in this layer. So how is melanin synthesized? Okay, review. Anybody? How is melanin synthesized?
Hi, Doc. Good
Hello,
morning.
Doc. Oh,
Come again? Yes, go ahead.
Okay. Melanin is actually synthesized from the hydroxylation of tyrosine to dihydroxyphenylalanine or DOPA,
Okay, by what enzyme?
tyrosinase, Doc.
Okay, then from DOPA, it is converted to melanin. Okay,
Yeah.
so these melanocytes, thank you. So these melanocytes will donate the melanin pigment to neighboring keratinocytes. Okay, so at this layer, you have the structures found here, or the cells present here are your keratinocytes, melanocytes, and also some tactile cells for the Merker cells. Okay, then the next layer is this layer, the stratum spinosum. So in contrast to your stratum basale, which is composed of a single layer, this is composed of about 3 to 5 layers of cells. Okay, and the keratinocytes here are larger and polyhedral, but they become slightly flattened as they move towards the surface. So for this layer, what is the distinguishing characteristic? There is John. Yes. Presence of what?
Cytoplasmic processes, Doc.
Okay, so you have these short processes, short spines or prickles found on the surface of the cells, thus giving them a prickly or a spiny appearance. So you can also call your stratum spinosum as a prickle cell layer or spinous layer. So this is composed of short cellular extensions made of tonofibrils, and these short processes will connect or attach with the short processes of adjacent or neighboring keratinocytes to form the intercellular bridges. Okay. So in areas that are subjected to continuous frictions or pressure like the soles, you will have a thicker stratum spinosum, and it also contains abundant tonofibrils and desmosomes. Okay, then next we proceed to the third layer, which is the stratum granulosum. Okay, so this is the most superficial layer, which is of non-keratinized portion of the epidermis. It is composed of about 1 to 3 layers of cells, which are flattened or polygonal cells undergoing terminal differentiation. So for this layer, what is the characteristic? Anybody? For the spinos, you have the short spines or the prickle. For granulosome, what you see in the cytoplasm?
presence of keratohyalin granules, Doc.
Okay, so the intensely basophilic staining granules known as the keratohyalin granules, okay, which contains cysteine-rich and histidine-rich proteins, which are the precursors of the protein filaggrin or the filament aggregating proteins. So these proteins will aggregates or holds the keratin filaments together within the cornified cells of the stratum corneum. Okay, and then, so very obvious, no, it's very easy to identify this layer. Next is we have seen only in thick skin is the stratum lucidum. So although this is difficult to identify with the use of a light microscope since the cells here stains poorly. Okay, so this is considered as a subdivision of your stratum corneum. Okay, and this is described as a thin translucent refractile layer of 4 to 6 rows of very flattened cells, okay, which are keratinized cells. And here, the cells have lost their nuclei and other cytoplasmic organelles. So what is present in the cytoplasm are the closely packed or the densely packed keratin filaments. Okay, take note, this has lost its nuclei and cytoplasmic organelles. Then lastly, the most superficial, the outermost layer, the thickest layer of the epidermis is the stratum corneum. So this is composed of about 16 to 20 layers of cells. And the cells here are flattened, unnucleated, desiccated, or non-nucleated keratinized cells. Okay. So again, just like in your stratum lucidum, the cells have lost their nuclei and and other cytoplasmic organelles, and the cytoplasm is filled entirely with keratin filaments of soft type. Okay. Now the cells present here have a thick plasma membrane, okay, and it is coated with extracellular lipid, a layer of lipids that will form part of the water barrier in the epidermis. Now, in the uppermost or the superficial layer of the stratum corneum, layer here that will constantly loosen and it will desquamate, flake off, or slough off is known as your stratum disjunctum. Okay, so the thickness of your stratum corneum will then constitute the principal difference between thick and thin skin. So again, it's thicker in areas that are subjected to friction, frequent friction, pressure, abrasion, just like the calyces of the palms and of the sole. Okay, any questions so far? Okay, so I'll proceed to the second main layer of the skin, which is the dermis. Okay, so the dermis, this is composed of dense connective tissue, and it has two layers. So the outer, more superficial layer is known as the papillary layer. Okay, and this contains, or it consists predominantly of type 1 and type 3 collagen fibers. There are also elastic fibers present here. Now, in this layer, you will find numerous blood vessels that will provide the nutrition to the epidermis, and also numerous sensory nerve endings or nerve processes. Now, the papillary layer forms a major part of these dermal protrusions, known as the dermal papillae or papilla. Okay, these are finger-like protrusions that will project into the undersurface of the epidermis. And its counterpart in the epidermis is the epidermal peg or the red ridges. So together, this forms the dermal epidermal junction or interdigitation. Okay, and this will make a sharp demarcation between the dermis and the epidermis. So it is an even boundary. It forms uneven boundary or irregular surfaces. Okay, so by the way, in the papillary layer, this is more cellular than the reticular layer or the second layer of the dermis. So here you will find numerous fibroblasts, macrophages, and mast cells. So for the second layer of the dermis, which is the reticular layer, this contains, by the way, in the papillary layer, this contains loose areolar connective tissue. And in the reticular layer, this contains dense irregular connective tissue and also contains predominantly type 1 collagen fibers as well as elastic fibers. Okay. Now, these fibers will are oriented to form regular lines of tension known as what?
Langer's
lines.
Okay, what is the importance of Langer's line?
The skin incisions made parallel to these lines will heal faster and without scarring photo.
Okay, so that's why when you make incision, especially in the facial area. So the incision you have to make should be oriented to the natural orientation of the collagen fibers. It should be parallel to the natural orientation of the collagen fibers so that it will heal with least scarring. Okay, so within the reticular layers, you will find here structures present here would include your hair follicle, sebaceous and sweat glands, and also your sensory nerve endings. Okay, so I think that's all for the dermis. Okay, so present also in the skin are the sensory receptors and motor nerve endings. And the common receptors that we can find here that we can usually identify in our slides are the Pacinian corpuscles. So I am sure you have encountered this in the nervous tissue, right? Have you? Okay, this
Yes,
came out
Doc.
in the exam.
Yes, Doc.
So how do you describe the Pacinian corpuscle? This is one
Onion-like?
of, yes, go ahead.
Onion-like appearance with several concentric lamellae, Doc.
Okay, so just like the osteon, di ba? You have concentric lamellae. This is large ovoid structure found in the deeper part of the dermis and also in the hypodermis. So this is composed of concentric cellular lamellae, and between the lamellae are the lymph-like fluid. So this is the receptor for pressure and vibration. By the way, this is one of the easiest structure that you can identify during the histopractical exam, right? So aside from your adipose tissue, so just remember the characteristic onion-like appearance of this corpuscle. Okay, that's the Vater-Pacinian corpuscle. And then another nerve ending, encapsulated nerve ending, found specifically in the dermal papilla is the Meissner's corpuscles. So this is described as a tapered cylindrical structure that is oriented perpendicular to the surface of the skin. Excuse me. And this is receptor for what?
For light touch, Doc.
Light touch and
Again, this is for
sensitivity.
light touch. Okay. And then lastly, we have the Ruffini corpuscle. Okay, least identified in our slides. Okay, however, this is characterized as the simplest encapsulated mechanoreceptors in the skin for skin stretch and torque. So it has an elongated fusiform shape with thin connective tissue capsule. Okay, so I think that's all for the skin. Any questions before I proceed? Okay, so we now go to the skin appendages and its derivatives. So one of that is hair. So do you remember this cartoon character? She has the longest hair in Disney World. So this is Rapunzel. Okay, so the hair or the hair follicle is a keratinized structure which is derived from the tubular invagination of the epidermis. Okay, and it is composed of three segments. The first is the infundibulum. So this extends from the surface opening of the hair follicle down to the level of the opening of the, what's this? Sebaceous gland. Okay, and from this level down to the insertion of the erector pili muscle is the isthmus, the middle portion of the hair follicle. Okay, and the most lowermost portion, which is the inferior segment, where it will expand to form the hair bulb. Okay, between the isthmus and the inferior portion is the follicular bulge. So this is located near the insertion of the erector pili muscle, somewhere here. This is the muscle, and this contains the epidermal stem cell. Okay, so this is a cross section of the hair. Okay, and we have the following layers. So from the innermost or the central portion is the medulla. So how do you describe this layer? Where is James? Malapitan.
Yes, Doc.
Yes, James. So how do you describe the medulla?
The medulla, Doc, is composed of the closely packed nucleus, Doc.
Come again?
It is composed of the closely packed cells, Doc. Not sure.
Uh-huh. What
Yes, Doc.
else? What type of keratin is present in the medulla?
I'm not sure about this one, Doc.
Okay. So forming the central part of the hair is the medulla, but this is seen only in thick hair. Okay. So this contains large vacuolated cells containing soft keratin. Okay. Then peripheral to the lateral to the medulla, or its periphery, is formed by the cortex. Okay. So this is the largest or the main bulk of the hair shaft, and it contains most of the pigment of the hair. So this contains tightly compact cuboidal or flattened cells, and which are heavily keratinized. So for the cortex, it contains the hard keratin. Okay, and the most superficial or the outermost layer is the cuticle. Okay, so this is composed of squamous cells forming the outermost layer of the hair shaft, and this consists mostly of heavily keratinized cells. And the cells here are arranged in an overlapping pattern where their edges point upward. Okay, now in the deeper part of the hair, it is covered by a root sheath. Okay, so we have the internal, we have here the internal and the external root sheath. So the internal and external root sheath are from the epidermal root sheath, okay, whereas the outermost layer here is the connective tissue root sheath, comes from the dermis. Okay, here. So for the internal, the inner root sheath, okay, it has 3 layers. So the innermost layer is known as the cuticle. So this is similar to the cuticle in the hair cuticle, which is composed also of squamous cells, but their edges, their overlapping edges, are pointing downward. Excuse me. Okay, and then the middle layer of the internal root sheath is known as the Huxley's layer, which is composed of about 1 to 2 layers of flattened cells. Okay, and the outer layer is a single layer of cuboidal to elongated cells. Okay, this is known as the Henle's layer. Okay, so these cells here are in close contact with the cells of the external root sheath, okay, which is also part of the epidermal root sheath. So the external or the outer root sheath here are made up of unspecialized epidermal cells. Okay, it is made up of a single layer of cells in the deeper portion, but then it becomes stratified towards the surface. Okay, what about? Okay, and this fibrous sheet here, okay, this is the condensation of the fibrous connective tissue of the dermis. So this forms the dermal root sheath. So it forms the outermost covering of the hair follicle. Okay, and separating the hair from the dermis is this thick basement membrane composed of non-cellular hyaline membrane, and this is known as the glassy membrane. So again, this separates the hair follicle from the dermis. So this is a long section of the hair, and again, you have the medulla located centrally, and peripheral to that or lateral to that are the cortex. Then you have the outer and the internal root sheath. Okay. Now, on the deeper, in the inferior segment of the hair, it will expand to form the hair bulb, and it will be invaginated with tuft of vascularized connective tissue, and this is known as the hair papilla. By the way, just to make a distinction between the hair papilla and the dermal papilla, in some books this is also known as the dermal papilla. Okay, but during the practical exam, when you are asked to identify this structure, then this should be hair papilla. Understand? Is that clear?
Yes, Doc.
Yes,
Doc.
Doc.
Yes, Doc.
Yes, Doc.
So if we answer dermal papilla during the practical exam, that is wrong. We should identify this as hair papilla, just to make a distinction between the hair papilla found in the hair follicle and the dermal papilla that is found in the papillary layer of the dermis. Okay, I hope that is clear. Okay, and then in your hair papilla, okay, this is surrounded by a region known as the bulb matrix. Okay, the bulb matrix, which is composed of dark staining mass of cells, okay, immediately adjacent to the hair papilla, and this is the matrix cells or the hair matrix. So this is the germinative layer of the hair follicle. So therefore, this is responsible for the growth of hair. Okay, so what else? I think that's all. Okay, important structure that is associated with the hair is this tissue, the erector pili muscle. What type of tissue?
Doc, a
muscle
smooth
tissue.
muscle tissue.
Smooth muscle, Doc.
Okay, so identify tissue that is smooth muscle. Okay, so this is attached midpoint in the hair follicle at the isthmus to the dermal papilla. So what is the importance of this muscle?
Doc, but ako po,
Yes,
what it does is it would, when it contracts, when it receives signals from the sympathetic nervous system, it would contract and it would, di ba naka-oblique angle man si hair. When it contracts, mag-vertical na ang hair. And also, it would help squeeze out secretions from the sebaceous gland po. And also, because nag-erect na ang, nag-vertical na ang hair, it would help with, this is what you call the pillow erection. So what it does is, in animals, it would help with heat reservation.
Okay. So
Ay,
especially
heat conservation po. But in
conservation.
humans po, it doesn't really help, but it would just form goosebumps po.
Okay, okay, thank you. So again, during in cold environment, okay, this will contraction of this muscle will pull the hair shaft to a more erect position in order to trap a layer of warm air near the skin. Excuse me. Okay. Okay. Then we proceed to another appendages of the skin, which are your sweat and your sebaceous gland. Okay, for the sebaceous gland, okay, this is your sebaceous gland, no? So this gland is associated with the hair follicle, very obvious. Okay, so except, no, this is not found in the palms of the the palms of the hands, sorry, and soles of the feet, wherein you don't have hair follicles there. So this sebaceous gland will form part of the pilosebaceous unit. And what are the structures comprising this unit?
Doc, you have the sebaceous gland, the hair follicle, and the erector pili muscle.
Okay, so, and sebaceous glands, it has highest concentration in the face. There are specialized sebaceous glands, meaning these glands are not associated with hair follicles. And so they open directly, or they kind of release their secretion directly on the surface of the skin. So can you give me one specialized sebaceous gland?
Montgomery's gland, Doc.
May bone yan, Doc, in your eyelids, Doc.
Okay, so
Then you
have,
also have Zeiss in eyelids as well, Doc. Then Tyson's in your glans penis, Doc.
okay, where else? Also found in the vermilion border of the lips, okay, as well as in the areola. Okay, so again, this develops as an outgrowth of the external root sheath of the hair follicle. Now, what is the major cells present in this gland? The predominant cells.
Sebocyte, Doc.
Okay, and so these sebocytes will produce and accumulate the fatty, the oily kind of mixture of the sebum. Okay, so what is the anatomical or the morphological classification of your sweat gland? Anatomical or morphological classification. Still remember?
Simple asigner, Doc.
Is this the simple branch
Simple
assigner?
This is simple branch assigner or succular glands. If it's larger, then it's succular. Okay. And then based on gland cell participation in secretion, do you classify this gland?
Holocrine secretion.
Why holocrine? Why not
Is it not
merocrine?
is it not merocrine, Doc?
Merocrine. Diba?
Which is
Kasi
which?
sa merocrine manggod it just it would secrete via cytosis lang and doesn't really bring out the part of the cell or cell membrane.
What about the others?
Holocrine, Doc.
Why holocrine?
Okay, during its excretion of the hormone, the secretion, Doc, the cytoplasm will break out.
Okay, so actually this is holocrine type gland. Holocrine, why? Because when the cells will undergo complete destruction during the process of secretion, that is holocrine. Okay, and they will constitute the secretion of the gland. Okay, the cells will undergo programmed cell death or apoptosis. Okay, and then since during puberty there is increased production of there is increased activity of the sweat glands, therefore there is increased production of sebum. This is in conjunction with increase in the level of what hormone?
Testosterone, Doc.
What else? What other hormones do you have? Specifically, what? Testosterone or your androgen. Okay, increase in your androgen level. So therefore, during puberty, you will notice na the breakouts on the face, especially on the face because of the higher concentration of these glands on the face. Okay, with the increase in secretion and alteration of the lipid composition, plus the action of your propionibacterium, acne bacteria, so it would lead to the formation of acne vulgaris. Okay. This gland has a secretory portion, which is made up of this round sac and a short duct that opens directly into the lumen of the hair follicle. Okay. Okay. Then the next gland, we have the sweat glands. So what is the, it is composed of two. You have your eccrine and your apocrine sweat glands, classified rather into two, into eccrine and apocrine sweat glands. So for your eccrine sweat glands, these are the glands that are not associated with hair follicle. So what is the anatomical classification? Diba? For your sebaceous, it is simple branch, acinar, or succular. How about for the eccrine sweat gland?
Coiled tubular, Doc.
Simple coiled tubular
Simple
glands.
Coiled tubular.
Okay, it's simple coiled tubular glands. Okay, and then most of these are located on the palms, the hands, soles, the forehead, as well as in the trunk. Okay. So the major role of your sweat glands is for temperature regulation. Now it also has secretory segments, which are located deep in the dermis or in the upper part of the hypodermis. And the secretory segment is composed of three cells. Namely, you have the light cells here, okay, which contains abundant glycogen, and the dark cells, which contains abundant rough endoplasmic reticulum and the cis secretory granules. Okay, located at the basal part. Where is the basal part? Here. The basal part of the secretory segments are the myoepithelial cells. So this contains the contractile filament actin. So therefore, when it contracts, it releases, or this is for the rapid expression of the sweat from the gland. Okay, for the dark segment of the eccrine sweat glands, okay, the dark segment here. So this is less coiled and a direct continuation of the secretory segment, and it leads directly into the epithelial surface. Now, in the dermal portion, the excretory or the dark segment is lined by stratified cuboidal epithelium. But then, when it reaches the epidermis, Okay, so the duct cells will end here in the epidermis. So this will be lined by the epidermal cells. Okay, at the level of the epidermis, it will take a tighter spinal force, forming cleft-like channels known as corkscrew channels or your sweat pore. Okay, so at this level, it is lined by the concentrically arranged epidermal cells. Okay, is that clear? So these are your corkscrew or your sweat pore. Okay, then the next is the apocrine sweat glands. So how does it differ from the eccrine sweat glands? Anybody? What is the most obvious difference between apocrine and eccrine sweat glands structurally?
Wider lumen, Doc.
Okay, it has a larger, wider lumen as compared to the eccrine sweat glands. And these glands also are associated with hair follicle. Okay, so they release their secretion into the hair follicle at the level just above the entry of the sebaceous duct. And then for the eccrine sweat glands, these are located so it is located widespread, so any parts of the body. However, for the apocrine, it has localized. No? So where can you find apocrine sweat glands?
In the areola of the breast, Doc.
Okay, where else?
In the mons pubis, Doc.
Mhm.
Doc.
Okay, usually in the found in the axilla, the areola, and the nipple, and also the skin around the anus and the external genital area. Okay, so it also has a secretory segment and the duct segment. Okay, for the secretory segment, it is composed only of a single type of secretory cells, which are either cuboidal or columnar. Okay, whereas for the eccrine, it is composed of you have two secretory, the dark and the clear cells. Okay. And also, you will have the myoepithelial cells, okay, located also in the basal part of the secretory segment. So again, contraction of these myoepithelial cells will bring about the release of the secretory products. Okay, and then for the duct segment, it's similar to the duct segment of the eccrine gland because it has a narrow lumen. It is also lined by stratified squamous cuboidal, and it lacks myoepithelial cells. Okay. Okay, so any questions before we proceed to the nails? By the way, the eccrine sweat glands, the onset of function is since birth, whereas for the apocrine, it starts to function at puberty. Okay. The nature of secretion of your eccrine is watery, whereas for the apocrine is viscid, which is rich in protein. Okay. For the eccrine glands, it opens directly onto the surface of the skin via the sweat pore or the corkscrew channels. But for the apocrine, it opens into the hair follicle since it is associated with the hair follicle. Okay, so next we have the nails. Okay, I'll just make this fast. So for the nails, this is a cornified plate that covers the dorsal surface. Now covering the dorsal surface of the distal phalanges of the fingers and the toes. So this is made up of keratinized dead cells of hard keratin type. Okay, so we have the free edge here, which is the distal part, and it's free of the nail bed. Then the visible part is known as the body. Okay, this covers and is strongly attached to the underlying nail bed. Okay, and the most proximal portion is known as the root, the nail root, which is covered by a skin fold. So this covers the cells of the germinative zone or the matrix. Okay, and this whitish crescent-shaped portion here is known as the lonula. So the lonula is best seen in your thumb and in your big toe. Okay, so this demarcates the distal portion of the germinative zone. Okay, and the color here, why is the color here whitish? Anybody? So the color here is derived from the thick opaque layer of partially keratinized matrix cells. Okay, so this is the nail bed. Okay, so histologically, this is how the nails would look like. The nail bed, which is covered by the nail plate. So this nail bed is composed of skin, which is made up of the Malpighian layer of the epidermis. What comprises the Malpighian layer of the epidermis? Any idea? Okay, this is composed of the first two layers, the stratum basale and the stratum spinosum. Okay, and the nail plate here is composed of closely packed interdigitating corneocytes, which lacks nuclei and other cytoplasmic organelles. Okay, in the proximal part of the nail, you have there the nail matrix or the germinative zone. Okay, so this is the one where the formation of nail substances will occur, and this contains numerous cells, one of which is your stem cells, okay, that will regularly divide and migrate towards the root of the nail, and here it will differentiate and produce the hard keratin of the nail. So for hard keratin, they do not disformate, okay, and it contains a high sulfur, which is responsible for the hardness of the nail. Okay, at the edge of the skin fold covering the root of the nail, okay, the root part here, is known as the eponychium, or otherwise known as the cuticle, and this is composed of hard keratin. And opposite to that, on the distal portion, is a thickened keratinized skin beneath the free edge of the nail plate, and this is known as the hyponychium. So this will secure the free edge of the nail plate at the fingertip or in the toes. Okay, I think that's all for the nails. Any questions? Okay, it's already time. So this is my last topic on the mammary gland. So another appendages of the skin are the breasts or the mammary glands. So this develops under the influence of sex hormones. So you have your testosterones for the males, which inhibits further growth of the mammary gland at puberty. And for the females, you have the estrogen and progesterone. So mammary glands are classified as modified tubulo-alveolar apocrine sweat glands. Okay, so this lies within the subcutaneous tissue. Now, in an inactive mammary gland, this is composed of about 15 to 20 irregular lobes separated by fibrous band of connective tissue. Okay, and these lobes will further subdivide to form the lobules. Take note that for the inactive mammary gland, this is composed largely of fibrocollagenous connective tissue. Okay. Then in contrast to that, for the lactating mammary gland, this is composed of small or grape-like clusters of small secretory alveoli. Okay, so these are seen they have well-developed alveoli. Alveoli are not found in a resting or an inactive mammary gland. So these alveoli are well-developed and branched from the duct system. It is lined by a single layer of cuboidal epithelium, okay, which are distended or it's partially filled or fully filled with milk. Okay, now the alveoli and the lobules will expand at the expense of the connective tissue. That's why in a lactating mammary gland, there is a reduction in the amount of connective tissue. Okay, for the duct system of the mammary gland, we have here the glands will end, each glands will end in a lactiferous duct. Okay, just like this. This lactiferous duct will open into the constricted orifice of the nipple. And at the base of the nipple, the lactiferous duct has an expanded or dilated portion known as the lactiferous sinus. Okay, so from the lactiferous sinus, it will give rise to the lobules beneath the lobular duct system beneath the glands. Okay, so your lactiferous duct is lined by what type of epithelium? Obviously, no? Stratified squamous epithelium. However, the lactiferous sinus is lined by what type of epithelium? Cabrera, Jacob. The expanded
no.
portion, yes.
Uh,
Is it still lined with stratified
no,
squamous?
I don't think so, Doc.
So what is the lining epithelium of the lactiferous sinus?
I'm not really sure here, Doc. Is it like is it like a connective one? I don't know.
Yes, okay. So it is lined by a single layer of either columnar or cuboidal cells. Okay.
Okay, Doc.
See, okay. Okay, lastly, for the terminal duct lobular units, as mentioned earlier, the duct system begins in the nipple from the lactiferous duct. Okay. And then from here, the lactiferous duct will be expanded at the base of the nipple. It has an expanded dilated portion, which is known as the lactiferous sinus. Now, this sinus will divide repeatedly to form the terminal ducts, okay, that will open into a lobule, okay? And the duct and the lobules here comprises the which is composed of multiple acini or ductules. So the terminal ducts with their associated lobule forms the terminal duct lobular units. So this is seen in a lactating mammary gland. Okay, so I think that's all. So are there any questions? Okay, if you have questions, then you can post it in our Histo classroom. So don't forget this afternoon, you will have the histo exercise on the skin. Okay, so if there are no more questions, clarifications, then we can go.
Excuse me, Doc. What time will you be releasing the activity for this afternoon?
For this, okay, I'll release it at 12 noon. Anyway, it's just a very short exercise. So I hope nobody will be late. So I'll try to release it at 12 noon.
Thank you, Doc.
Okay. Okay, so thank you, everyone.
You, Doc.
Thank you, Doc.
Thank you, Doc.
Good.
Thank you, Doc.
Thank you, Doc.